Bismillahirrahmanirrahim. I am Dr. Rabia Yaqoob from Department of Zoology, Division of Science and Technology, University of Education, Lahore. The work I'm going to share with you today is about assessing the anti-inflammatory role of peptides extracted from venom of butthead scorpions. Scorpions are the oldest arachnids which are widely distributed over different land habitats. There is a huge diversity among various kinds of scorpions, and Buthidae is the most abundant and most venomous family, comprising of 89 genera with 1,954 species. Common Buthid genera, which were found in the surrounding areas where the study has been conducted, are Androcnus, Mesobuthus, and Odontopothus. Normally, scorpion venom is considered as toxic substance which is associated with induction of severe pain. But there are some specific peptides which are proved to be the potent healers of various maladies. These peptides could potentially be used in the physiological and pharmacological research, as well as they can also be used as investigatory research tool. Peptides of low molecular weight are of utmost medicinal importance. These peptides may activate or inhibit activity of various ion channels, acetylcholine receptors, as well as coagulating and anticoagulating pathways. Normally, scorpion envenomation involves B lymphocyte-mediated activation of immune system, T lymphocyte-mediated activation of immune system, as well as upregulated production of catecholamine, angiotensin II, glucagon, cortisol, and nitric oxide. Cytokines produced after envenomation, they exert hormone-like effect on other cells and mediate inflammatory process, immune response, as well as they also speed up the tissue repair and healing process. Usually, cytokines are divided into two groups, pro-inflammatory cytokines and anti-inflammatory cytokines. Actually, anti-inflammatory cytokines resolve the hyperactive inflammatory process. Previous studies has shown elevated cytokine levels like interleukin-4, interleukin-6, and interleukin-10 after scorpion envenomation. So keeping in view the previously reported data, this study has been designed to isolate different peptide fractions of venom, scorpion venom, to evaluate the anti-inflammatory potential of selected peptides in mus musculus, to study the effect of crude venom and selected protein fractions on various blood parameters of mus musculus, as well as to estimate and compare the level of anti-inflammatory cytokines in the mus musculus. Talking about the materials and methods which were used in this study. First of all, scorpions were collected from the district Sargodha. For the scorpion collection, UV lamps were used and scorpions were collected during nights of the full moon night. Afterwards, the scorpions were brought to the laboratory in the Department of Zoology, University of Sargodha, where scorpion extraction was carried out. Venom extraction was carried out. For venom extraction, the venom gland was electrically stimulated and the venom droplet was collected in the vials. Then from that collected venom, isolation of peptide fraction was done by using polyacrylamide gel electrophoresis. For evaluation of anti-inflammatory potential of venom, study was divided into different parts. First of all, the determination of median lethal dose of the venom was done for mice. For this, intravenous injection of different venom doses was carried out. Then for evaluation of anti-inflammatory potential of venom, that was assessed by induction of paw edema. And the severity of paw swelling at ankle joint was assessed. Afterwards, the various blood parameters were evaluated. The inflammation was induced in mus musculus by injecting 1% carrageenan suspension. In the third part of experiment, effect of venom on the cytokine level was checked, and the level of interleukin-4, interleukin-6, interleukin-10, and interleukin-13 in the mice serum was assessed by using specific kits. For analyzing the effect of venom on the blood parameters as well as anti-inflammatory cytokines in the serum, different groups were formulated. In the negative control, we have injected 550 microliter of normal saline, and afterwards 50 microliter of 1% carrageenan suspension was injected. To the positive control, 50 microliter of betamethasone was injected, and afterwards 50 microliter of 1% carrageenan suspension was injected. And in experimental group, 50 microliters of sublethal doses of crude venom from different species as well as peptide fractions which are extracted. They were injected, and later on 50 microliter of 1% carrageenan suspension was injected. 1% carrageenan suspension was injected to induce the inflammation in the mice. As per as statistical analysis which are applied are concerned, normality test, descriptive statistics, analysis of variance, followed by Stuckey's test for multiple comparisons, and probability analysis were applied. Study was conducted at Department of Zoology, University of Sargodha, and Department of Zoology, University of Punjab, Lahore, where all facilities were available. Furthermore, the study was supported by, it was funded by the Higher Education Commission. Now, coming towards the findings of the study, first of all, the different fractions which were extracted from the venom, they includes AF1, that is fraction 1, containing the 54 kilodalton peptides from the venom of Androcnus finitimus. MF1, that contains the peptide fraction of size ranging about 54 kilodalton from the venom of Mesobothus tamulus. OF1, it was the fraction of peptides of size 54 kilodalton from the venom of Odontobothus odontorus. AF2 contains the peptides of 36 kilodalton from the venom of Androcnus finitimus. MF2 contains the peptides of about 36 kilodaltons from the venom of Mesobothus tamulus. And OF2 contains the peptides of about 36 kilodaltons from the venom of Odontobothus odontorus. Likewise, AF3 contains the peptide fraction of about 8.5 kilodalton from the venom of Androcnus finitimus. MF3 contains the peptide fraction of about 8.5 kilodalton from the venom of Mesobothus tamulus, as well as OF3 contains the peptide fraction of about 8.8 kilodalton from the venom of Odontobothus odontorus. In the first part of experiment, LD50 was found for different venom of different scorpion species. This graph is showing the LD50 of crude venom of Odontobothus odontorus. That is about 0.0299 milligram per gram of animal. This graph is showing the LD50 of crude venom of Mesobothus tamulus, which is 0.0375 milligrams per gram of animal body weight. This graph is showing the median lethal dose of crude venom of Androcnus finitimus, that is 0.019 milligram per gram of animal bodies. These results shows that the Androcnus finitimus venom was the most toxic because it has the least LD50 value. These graph shows the effect of venom on carrageenan-induced paw edema in mice. Injection of carrageenan result on a remarked swelling of tibiotarsal joint within 1 hour of injection. Past swelling in negative control animal increased progressively up to 3 hours post-treatment and then decreased gradually, but do not return to normal size even after 12 hours. When the effect of Mesobuthus tamulus venom and its protein fraction on carrageenan-induced paw edema was recorded, decline in paw edema swelling was observed in all treated groups as compared to negative control. Recovery was most rapid in MF3 fraction treated groups as paw size returned to its normal size during 1 hour post-treatment. In OF1 treated animal, rapid decrease in paw swelling was observed during first hour of treatment, followed by OF2 and OF3 treatment. While in OCV, that is crude venom of Androcnus, treated group recovery in paw swelling was slow during 2 hours post-treatment. Increase in paw diameter or paw swelling was observed in ACV and AF1 treated animals during first hour after treatment with which resolved within 4 hours post-treatment. Increase in paw size was more obvious in ACV injected group as compared to F1 treated group. Animals treated with AF2 and AF3 peptide fractions had reduced paw swelling during first hour of treatment, and resumption in paw size was more apparent in AF2 treated groups. This table shows the effect of Mesobuthus tamulus venom and its protein fractions on different blood parameters. You can see that total leukocyte count was maximum in MF1 treated groups, and there was a significant difference among different treatments. There was a significant difference in the total leukocyte count in different groups. As far as neutrophil count is concerned, that was count as percentage of total leukocyte. Highest amount of neutrophils were observed in MF2 treated groups, and there was a significant difference among various treated groups. In case of lymphocytes, the maximum number of lymphocytes were observed in the MF1 treated groups. As far as monocytes are concerned, MF1 and MF2 treated groups showed the higher level of monocytes. Eosinophils eosinophil levels were most elevated in MCV, MF1, as well as MF3 treated groups. As well as RBC count is concerned, it was maximum in the MF3 treated group. And the hemoglobin concentration that was calculated as grams per deciliter, it was maximum in MF3 treated group. And there was also significant difference among different groups, the different treated groups. This table is showing the effect of Odontobothus venom and its protein fractions on different blood parameters of mice. You can see here that the total leukocyte count was maximum in OF3 treated groups. Neutrophil count was maximum as OF3 treated group, and lymphocyte count was maximum in OF1 treated group. As far as monocytes are concerned, their count was maximum in OCV treated groups, and the eosinophil count was maximum in OCV treated group. Total RBC count was observed maximum in OF3 treated group, as well as the hemoglobin concentration was maximum or highest in the OCV treated group. This table is showing the effect of Androcnus finitibus venom and its protein fractions on different blood parameters of mice. You can see here that AF3 fraction of Androcnus venom elevated the most total leukocyte count. As far as neutrophils are count, they were also maximum in AF3 treated groups. Lymphocytes were maximum in AF2 treated groups. Monocytes were maximum in all AF1, AF2, and AF3 treated groups, while eosinophil count was maximum in ACV and AF1 treated groups. RBC count was highest in ACV treated group, while the hemoglobin concentration was maximum in ACV treated group. Although there was no significant difference among the monocyte count, RBC count, and hemoglobin concentration among different treatments of different peptides, peptide fraction or crude venom. This table is showing the dynamics of anti-inflammatory cytokines in serum of mice, which was injected with whole venom and various venom fractions of Mesobotus tumulus. These values are that are represented in the table. They indicate the absorbance at 450 nanometer. These are the absorbance values at 450 nanometers. Interleukin absorbance for interleukin 4 was highest in the MF2 treated groups, and there was a significant difference among different treatment groups. As far as interleukin 6 is concerned, its value was maximum in MF3 treated groups. Interleukin 10, in the case of interleukin 10, absorbance was maximum in MF1 treated groups, while interleukin 3 shows the maximum absorbance in MF3 treated groups, and there was a significant difference among with different treatments. Table 5 is showing the dynamics of anti-inflammatory cytokines in serum of mice injected with whole venom and various venom fractions of Odontobothus odontorus. Here you can see that interleukin 4 concentration of interleukin 4 was maximum in OF2 treated groups. Interleukin 10 concentration was maximum in OF3 treated group, as well as the absorbance in case of interleukin 10, it was maximum in OF3 treated group, while absorbance in case of interleukin 13 cytokine was maximum in OF3 treated group. This table is showing the dynamics of anti-inflammatory cytokines in serum of mice that was injected with whole venom and various venom fractions of Androcnus finitimus. Here you can see that interleukin concentration was maximum in case of AF2 treatment. Interleukin 6 concentration was maximum in case of ACV treatment. Interleukin 10 concentration was maximum in case of AF3 treatment. and interleukin-13 concentration was maximum in case of AF3 treatment. And in case of all the interleukins, there was a significant difference among different treatments. So concluding the findings of study, we have found that crude androcnous spinetimus venom was the most toxic in terms of LD50. Total leukocyte count and differential leukocyte count decreased as a result of crude venom treatment. Overall increase in differential leukocyte count and total leukocyte count was recorded in F2 and F3 treated groups. F2 and F3 found to be more effective in increasing the levels of anti-inflammatory cytokines. Future perspective of study. As studied peptides are found to be effective in healing of inflammation, so we recommend further study on these peptides to modify them for synthesis of new and cheap drugs for curing edema or inflammation related diseases. Different fractions act in different manner. Use of venom peptides in different combinations yet to be evaluated to get maximum curative effects. Furthermore, sequencing of these separated peptides is required to get better insight of mode of action of those peptides. That's all. Thank you for your kind attention. Allah Hafiz.